All right, good day, everyone. Quickly, I'm going to talk about a simple aspect of molecular biology, which is termed genetics. Okay, I'm going to look at genetics today, and I'm going to do a lot about genetics. We're going to explain and see how we can um, relate the basic concept. Now, meanwhile, it should be noted that this video is going to come in two. In this first video, I'm going to do introduction to genetics. I'm going to explain the basic terminologies, the difference between the various terms in genetics and what they actually mean. So in the next video, we're going to do more of Mendelian inheritance. We're going to talk about Gregor Mendel, who's an Austrian monk that was the father of genetics, or popularly known as the father of genetics. He did a lot of research on genetics, and till today, some of his research are still very valid. So I'm going to talk about Mendelian genetics and non-Mendelian genetics. So there are also some genetical characters, also genetical properties of organisms today that Mendelian's theory could not explain. So in the next video, I'm going to look at that. But meanwhile, for this video, so that it will not be very, very long, I'm going to explain what's it called? I'm going to explain the basic term genetics. Now, what is genetics? Genetics is actually the study of hereditary, how materials are passed from parent to offspring, which is hereditary, the study of hereditary and variation. So variation is the differences that occur between living organisms of the same species. So, okay, you can get someone like me, that is someone human, Homo sapien, like I am. Is fair, me I'm dark. So there's a difference. So that's variation. So the study of hereditary and variation is referred to as genetics. Genetics was coined by the word genetics was coined by the man called William Bateson. William Bateson, the early 20th century. So William Bateson coined the word genetics. Okay, um, that's um, for the introduction of genetics. So what's genetics? What is genetics proper? Genetics has to do with mainly hereditary and variation. Now, the hereditary part, the variation part can come later. Okay, now for the hereditary part, hereditary is how characters are transferred. Characters are transferred from parents to offspring. So we cannot just know how characters are transferred from parent to offspring without knowing the basic terminologies in hereditary, without knowing the basic terms that the living organisms contain that can help to transfer these characters. You can see some diagrams, so you can see DNA, RNA, proteins. So I'm going to explain. Now, quickly, every organism have a basic unit known as the cell. Okay, so for instance, the animal cell, which has no cell wall. Now, the animal cell should have a very large organelle in it, which is called the nucleus. The nucleus has a dark spot that is highly pigmented, which is known as the nucleolus, and it has some chromatin thread in the making up the what they call the nucleosome. So the nucleus of every organism first is responsible for reproduction. It's responsible for reproduction of this cell, because in every organism, these cells are going to undergo division. These cells are going to multiply. In terms, as far as the organism grew, one cell turns two cell turns four cell turns eight, like that, and so on. These cells are going to multiply. So, but what will help to multiply is the nucleus. The nucleus plays an active role in that. Now, the nucleus is made up of two things. The nucleus of a cell is made up of two things. Okay, the nucleus of a cell is made up of two things. The first one is what we call the nucleolus. Nucleus made up of two things. The first one, I'll say the nucleolus, and what I call the nucleosome. Yes, the nucleolus, pardon me, the spellings, the nucleolus, and what I call the nucleosome. Okay, now the nucleolus helps is the setup and production of DNA, RNA, and co. So, but the nucleosome is very important, very, very important to cell division, because when the cell is about to divide, if you have read your cell divisions, when the cell is about to divide, this nucleosome is going to disappear. All right, pardon me, this nucleolus is going to disappear. So the nucleolus disappear during cell division. This is a very important part of prophase. the nucleolus will disappear. Okay, now remaining leaving just the nucleosome, which is actively involved in the division of the cell. Okay, now quickly. So how does it work? What does the nucleosome contain? The nucleosome contains things like the chromosome or the chromatin thread for a resting cell. Now, it contains substances like DNA and it contains substances like proteins, usually histone proteins. That's for the nucleosome. Yes. So the nucleosome is more like the materials in the nucleus that is not the nucleolus. Those are the nucleosomes. So they are basically these things and some other proteins and some other materials. So basically these ones. And they are all involved in the transfer of materials from one cell to another. So let's quickly go. Now, these are the substances in the nucleus. They will be involved in the transfer of materials from parent to offspring, also from one cell to another. So how do they do this? So we have to start taking them one after the other. So let's take chromatin. So let's look at the chromatin. So you might have to wipe this. So you can pause and look at what I just wrote. Now let's take, for instance, the chromatin. Yes, the chromatin. For instance, the chromatin. Yes, the chromatin. Now, the chromatin, it has, before I explain, chromatid, chromosome, and chromatid. Then I will explain the concept of gene. Now, quickly, we should know that every living organism have various types of cells. Living organism, before I explain this, so it will make more sense. Every animals have, let's use animals for instance, have various types of cells. So basically, I can classify the cells into two. I can have what they call somatic cell, or what we call normal body cells, and germ cells. Okay, germ cells are also called reproductive cells. Okay, what do reproductive cells do? Germ cells or reproductive cells. Now, what do they do? What do reproductive cells do? What do germ cells do? Remember, germ cells or reproductive cells, they also called reproductive cells. Now, what is somatic cell? What is a reproductive cell? Now, first, somatic cells are your normal body cells that will not be able to carry out reproduction. Cells of your hand, cells of your leg, cells of your eye, cells of the buttocks, cells of the gluteus, and so on. So those cells that cannot carry out, that are not involved in reproduction, usually somatic cells. Now, what are reproductive cells or germ cells? Reproductive cells or germ cells are actually cells that are involved in reproduction. Now, these cells undergo meiosis and mitosis, while somatic cell undergo only mitosis only. Yes, while this reproductive cell can undergo both mitosis and meiosis. Yeah, so when they undergo meiosis, one cell is going to, because in meiosis, obviously, they undergo meiosis, one cell is going to divide into four in most cases, into four, in which one of them could be more developed, like for the ladies or for the ovum cell in women or in girls. In the ovum cell, we have one cell divided into four, in which one of them will be the main cell, will be the main ovum cell, while others will be the secondary cells, or will form what they call the polar bodies. So you can look at that. It's called that this process I'm explaining is called oogenesis. You can check oogenesis out. Okay. Now for this particular cell, if one cell divided into four, for meiosis, let's say this is a reproductive cell, cells of the testes, okay, the Leydig cells. So if one of these cells divided into four, um, each of the four cells, this was a diploid. If this was what they call diploid number of chromosomes or diploid number of chromatic materials, this of these four cells is going to have haploid number. So every living organism in us, because I'm coming to explain what a chromosome is. So every living organism has 40, every human, every Homo sapien has about 46 chromosomes. Every Homo sapien, why for Canis familiaris, the dog has about 78. Drosophila melanogaster, fruit fly, has about 8 chromosomes. So, but for us, because obviously, if you're watching this video, you should be a man, should be a mammal, should be man, should be an Homo sapien. So we have 46 chromosomes, 23 in pair. So 23 pair making it diploid. So 23 pair. So, but once a cell, a reproductive cell undergo meiosis, as undergo meiosis, it's going to produce um, four other cells, each having only 23 chromosomes, not in pair. So this time not 46, not 23 pairs 46. Now each of them will have 23 chromosomes. So this is what meiosis results in. Now, but for mitosis, if a two-end cell, normally is going to divide into two. They're going to divide into two end, two end. So each of these cells are going to have what they call, are going to have the same number of chromosomes as the parent cell. That's for mitosis only. But for once a cell undergo meiosis, is going to be the number of chromosomes is going to be halved. So only reproductive cell are capable of undergoing meiosis. So meiosis, and these haploid cells can be used for reproduction. These haploid cells can be used for reproduction. So an haploid cell from the father is going to fuse with the haploid cell of the mother. They are usually called gametes. So haploid gamete fuse with the haploid gamete of the mother, haploid gamete from the father, haploid gamete from the mother, they join together, and when they fuse, they form a diploid zygote, which can grow through stages for mammals, through a stage, through some various stages such as gastrulation, organogenesis, and so on and so forth. So we'll look at those in subsequent videos. But this is how the two types of cells can undergo meiosis and mitosis. So let's see what is a chromosome. So when you are reading meiosis and mitosis, so when you hear the chromosomes number as half, or is now half, you know what it means. What is a chromosome? So you can see clearly that this chromosome can play roles in reproduction. So what is a chromosome? What is a chromosome? What is a chromosome? What is a chromosome? Now quickly, I'll pick chromosome. Now, a chromosome is actually a nuclear material found in every cell that can help to transmit materials from parent to offspring. All right. Now, the unit of a chromosome is a gene. The unit of a chromosome is called a gene. Okay, now let me explain. So these chromosomes in a cell, for instance, these are cells, nucleus, chromosome. Okay, this is chromatin. Okay, now these chromosomes are usually made up of some things. Now the things is made up of a long polymer called DNA. A long polymer called DNA that could be alpha helical in structure or beta plated in structure. Now, and it is also made up of some proteins, usually histone proteins, and some non-histone. So the proteins might not be histone proteins. Yes, these histone proteins occur in pairs of 8, called octamers. So, but DNA are made up of, or rather, chromosomes made up of a DNA and an histone protein, and some non-other histone protein. So DNA plus protein make up chromosome. The unit of the chromosome is called a gene. So let me just quickly make you understand what I'm trying to talk about. Okay, now the chromosomes are materials inside the nucleus, making up the nucleosome. Now they can replicate themselves. They can replicate. When they replicate themselves, they can pass information to another substance, which we normally call the RNA, to send information to other cells, to tell the cells these are the kind of information coming from the parent. So those are chromosomes. Now the chromosomes are made up of DNA, are made up of histone, are made up of non-histone. Now the main character or the main substances in the chromosome that is helping the chromosome to display information or send information to other parts of the cell to tell them what type of information is coming from the parent is the DNA. All right. So remember, I said the unit of the chromosome is called a gene. The chromosomes are made up of DNA, histone proteins, and non-protein histone. Okay. Now, the chromosome is made up of long molecule called DNA. Now, what is a DNA? A DNA quickly means deoxyribonucleotide. Deoxyribonucleotide. Then I'll have to comment. That's a DNA. Deoxyribonucleotide. Now, deoxyribonucleotide, DNA, is actually a long molecule, like you are seeing here. a very long molecule. So this is an alpha, and this is an helical form. So remember alpha helical, it can be better plated. So it could be something like this. Yes. Yeah, it could be something like that. So, but this is an alpha helix. So when it occurs like this, it's said to be alpha helical. Yeah. So it could be better plated like that. So, but this is an alpha helical form of a DNA. Now, this DNA is a long molecule, it's a polymer, and it has repeating units. It has repeating units. A polymer should have repeating units if you are good in chemistry. And that repeating unit is called a nucleotide. So I call that repeating unit a nucleotide. A nucleotide. So my spelling is called a nucleotide. So the repeating unit of a DNA is called a nucleotide. So DNA is a nucleic acid, the polymer long molecule. It has one unit called nucleotide. So many nucleotide will make up a DNA. Now, in this DNA, they can be very long like this. They can be very long, very, very long. Okay, let's make it longer. They can be very long. This DNA molecule can be very long, can be very long. Now, one segment of the DNA, it can be like 10 nucleotides, it can be like 6 nucleotides, it can be like 7 nucleotides. One segment of the DNA is known as a gene. So I've defined genes in two ways. I say genes are the unit of chromosome. So the repeating unit of a chromosome in a cell, each unit of the chromosome is called a gene. Now, I also said segment of DNA is what we call a gene. Now, let me make it clear. Now, on this board, now what I've just drawn here is a DNA, but I did something. Now, this DNA, this is a DNA, long DNA molecule. Let's say, assume it's longer. This can be a DNA. Now, if this is a DNA longer, now this DNA can have some proteins at the side of it. When it has proteins, these proteins helps to do a lot. This protein helps to do a lot in condensation, in coiling, in decondensation. This protein plays a lot of function to this DNA. So when it has a lot of proteins attached to it like this DNA molecule, rather, this DNA molecule with the proteins become a chromosome. It can condense, it can shorten. Well, we now have the likes of chromatin and chromatid. Okay, I will explain better. So it can shorten, it can condense, it can compress to form chromatin. So this DNA long molecule plus proteins becomes a, what's it called now? Becomes a chromosome. What I've just said is that if I now take a segment, let's say I take 3 nucleotides, I take a segment of this molecule having a long DNA and protein, which is called chromosome. If I take a segment of it, it becomes a gene. So genes are the unit of chromosomes, or they can be called segments of DNA because the chromosome itself are made up of DNA all around. Yes. So I will this the first person to quickly tell us more about how gene works was the man of the caliber of Gregor Mendel in 1856 when he studied serum sativum regarding P. Okay, so but that's that for that. That's that for that. So I'm going to let you understand it better and better and better. So that's for gene. So now quickly um um for the nucleotide, we already said the repeating unit of a DNA is called a nucleotide. So nucleotides are what is making up DNA. Okay, so this thing, this DNA molecule I drew here, this DNA minus the protein, now minus the protein, this DNA molecule I drew here, I expanded it here. So I expanded it here. So if you watch this very well, if you watch this very, 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 very well, you see this thing I expanded it. Now, the DNA molecule is made up of many nucleotides. Now, many nucleotides. One nucleotide is made up of this. Watch. One nucleotide is made up of a ribose backbone sugar, a nitrogenous base, nitrogenous base, which are of two, purines and pyrimidines, and we have a phosphate group, usually a phosphoric acid group. Yes, we have a phosphate group. So now the nucleotide consists of basically the ribose backbone sugar, the nitrogenous base, and the phosphate group. That's what the nucleotide consists of. Now, I therefore now open, so explaining the proteins, just the DNA molecule, DNA polymer. Look, I opened it here. So opening this guy here, I should have a ribose backbone, nitrogenous base. Now these bases normally are in pair, and we explain. And we have one, what is joining this part to the next is called the phosphate, to the next nucleotide is called the phosphate group. So this backbone sugar, this phosphate group, this backbone sugar, this phosphate group, backbone sugar, phosphate group, together is called a nucleotide. And it should be noted that now the nucleotide excluding the phosphate group is called a nucleoside. So therefore, a nucleoside is actually the ribose backbone sugar and the base pair. That's all for a nucleoside. All right, so this guy in bulk will make up in long molecule will make up a DNA, in which one segment, so one segment can be like 10 nucleotides, one segment of the long DNA is what we call a gene. So genes occur in pair. We're going to explain that still in this video. So now this is a nucleotide. All right. So now let me quickly explain what is the nitrogenous basis about. Now the ribose sugar is a 5-carbon sugar. For DNA to be very precise, the ribose sugar has lost oxygen at carbon 5. So it is called deoxyribose. Yes, deoxyribose, not just a ribose now, it's a deoxyribose for DNA. Why for RNA it's a ribose? So I'm going to explain the difference. Quickly. Now for basically this ribose is the backbone. It helps to hold the bases together. So remember, it helps to hold the bases together. And you must have heard that DNA is called the code of life. So DNA is, yes, it's the code of life. Why? Because it has nitrogenous base in it that are acting like code. Now, what do I mean? Now, for instance, we can say a DNA molecule should have AT, Now these are codes. AT, GC. So the nitrogenous base tries to code information as base pair. So it codes information as base pair. So when remember I said like 10 nucleotides makes up a gene. So 10 of these codes, so the code here, like 10 can be one gene. So this can be a gene for height. GC. Can be the code for height. Can be on one allele for height. So we have another one, 80 GC. So this is now what you will now be writing as BT. Yes, can be the code. And these codes are going to repeat for the long DNA molecules. They're going to repeat. And you can see more than one gene can be linked on a particular DNA. So that's that for that. So quickly, now DNA is the code of life because the bases are in code. It codes information as base pair, and it's going to replicate it. You can see some group can replicate it because of making the DNA or transcript it to RNA. It can transcript it to RNA, and that for that. So quickly, now, what is the base in a DNA? What's the base in nucleotide consisting? Now, the base in a DNA, the nitrogenous base can be two. It can be purine and it can be pyrimidine. So the nitrogenous base in DNA can be of two, purine, or it can be pyrimidine. Now, what are the purine bases? The purine bases are usually of two types. The purine bases are of two types. The first one is adenine for purine bases, and the next one is what's it called? Yeah, guanine. Yes, adenine and guanine. While for pyrimidine, there are three types of pyrimidine bases. Okay, we have thymine, not thymine like vitamin B1. Thymine, we have uracil and cytosine. Thymine, uracil, and cytosine. These are the nitrogenous bases. So where I represented them, A for adenine, and T for thymine, G for guanine, and C for cytosine. Now, quickly, it should be noted this. For a DNA molecule, for a nucleotide, many nucleotides makes up one DNA. So for a nucleotide, it should be noted that a purine in the base pair, a purine should bond with a pyrimidine. One purine should bond with a pyrimidine. For DNA, the base pair is adenine to thymine, while the other base pair is guanine to cytosine. Guanine must bond with cytosine. So if cytosine comes first before guanine, that's a change in code. Yes, that's a change. So assuming it was CG, it's a different code from GC. Yeah, that's how life is being coded. That's how life is being coded. So ATGC, that's how it is. So I should be noted that the bond between adenine, between the nitrogenous base, remember a purine should bond with pyrimidine. The bond between the nitrogenous bases, the bond between the nitrogenous base is known as hydrogen bond. So the hydrogen bond exists between the two nitrogenous base pair. So remember a purine should bond with pyrimidine. For DNA, A must bond with T, G must bond with C. So A bonding with T, you have a double hydrogen bond there. While G bonding with C, we have triple hydrogen bond for guanine to cytosine. So this bond pair is stronger. Guanine to cytosine bond pair is stronger. Now for the ribose backbone, the bond between the ribose and the guanine, is just a simple covalent bond. So the bond between the ribose and the base, the sugar backbone and the base, nitrogenous bond. While the bond between the ribose and the phosphate group, bond between the ribose and the phosphate group is called a phosphodiester bond. Yes, called a phosphodiester bond. So a phosphodiester linkage is the bond. Those of us in biochemistry, you know this very well. Now, the phosphodiester linkage is actually a linkage holding one nucleotide to the other. Please take note of that. So a nucleotide is held to another nucleoside through a phosphodiester linkage. That's for that. So that's for nucleotide. Nucleotide is a unit of the DNA. As recap, nucleotide, several hundreds, millions of nucleotides make up the DNA, while about 10 to 7 nucleotides is found in a gene, because just a segment of the DNA is called a gene. Okay, so that's that for that. So having known what is in the chromosome, having known what the chromosome is made up of, I can now talk about chromatin and chromatid. Now, and this chromosome has the ability to do something. Now, this very statement is what called the central dogma of molecular biology. Now, this is the direction or flow of gene. This is how gene is being interpreted in the new daughter. So, for instance, a parent gives birth to an offspring. These are the genes that are going to be interpreted. Yes. Remember, the genes are found in the DNA. Segment of the DNA is called the genes, and the DNA makes up the chromosome with some proteins. Now, this DNA can replicate themselves. So, inside a cell, a DNA can make another DNA. It's called DNA replication. It's a process. Maybe I'll do a video on that subsequently. So it's called DNA replication. In biochemistry, it's called DNA replication, where a DNA replicates itself, produces another DNA of the same kind. So when those two DNA are of the same kind, those two DNA are usually called homologous. They are usually found on homologous chromosomes. They are the same DNA. It's called DNA replication. Okay, so they replicate themselves and cross over at the centromere. I'll come and explain that. Okay, now when the DNA can also form the RNA. It's called DNA transcription, because the DNA can form, this is not called replication, can form an RNA. Yes, because the main difference between the DNA and the RNA, is their nucleoside. Now, the nucleotide of an RNA, the ribose is not deoxygenated, so we just have the ribose. Now, why also in the base pair, instead of having thymine for RNA, I have uracil. And there are three types of RNA. There's tRNA, there's mRNA, and there's rRNA. Okay, rRNA. So DNA can form RNA. There's an enzyme that does that. It's called RNA polymerase. RNA polymerase is involved in this process. So such that the information of the genes or the DNA are not transcripted to the RNA. So the RNA will send an information to the ribosome through the mRNA. So the mRNA does this now, send information to the ribosome, and the ribosome, with the help of the rRNA, is going to make proteins that are going to translocate, or they are going to interpret the idea of this RNA. Let's say the protein or the DNA of I use this as an example in class very, very well, just as a joke. For instance, when you have the genes of having big head, so genes of having big head. So the DNA is going to send information somehow by replication and transcription. It's going to send information to the ribosome. The RNA is going to translocate it to the ribosome. The ribosome is going to make proteins that will make you have big head. Yes. So that's how it works. It might not be visually how it really works, but that's the basic concept. So remember, this video is the basics of genetics. We're still looking at the basics. So if you have a question on this, you can ask in the comment section. These are the basics. So if there's any way you don't understand, you can take it back. The video is a little bit it has a lot of it. So in the next video, I want to look at medullary genetics. So that's for the 3 types of what's it called. We have 3 types of RNA, which I will come and explain later. So this is what we call the central dogma of molecular biology. It is how information has been carried from your chromosomes to each your individual cells to make yeah. So please now, that's how it is for that. Okay, that's for a chromosome. Now, chromosomes are only visible. It should be noted, a chromosome is only visible when a cell is about to undergo cell division. So normally, if I have a cell like this, I cannot see the chromosome. The chromosomes are highly invisible. Yes, highly invisible. So they are invisible. The chromosomes are condensed with proteins before cell division. When a cell is not undergoing cell division, the cell is said to be in an interphase state. So in interphase, the chromosomes are highly invisible. They are condensed. They are coiled to form a thread-like material called chromatin. So during interphase, that's where the DNA molecule are going to replicate and transcript. So yes, so they are going to replicate and transcript. All these processes occur during interphase. Okay, so remember I said the chromatins are they're invisible chromosomes. Okay, highly invisible chromosome in the nucleus during interphase, resting stage of the cell when it's not undergoing division. So the cell cycle is made up of 2 parts. There are 2 parts of the cell cycle. The life cycle of every cell is 2: interphase and cell division. Interphase involves 3 steps. 3 steps. Okay, so we'll look at that in cell division. Three steps. Okay, 3 steps: G1, G2, and the S stage. Okay, that's that for that. So now quickly, the chromatin are invisible. They are invisible. So when a cell is about to undergo cell division, this chromatin, when a cell is about to undergo cell division, and this chromatin will now condense to form chromosome. So remember, maybe I made a mistake. I don't know. Chromosome decondense to form chromatin. They decoy to form invisible chromatin. They decoy. So when the cell is about to undergo cell division, the chromosome will now be visible. So these guys are going to they're now going to condense to form chromosome. When they condense by changes. So these thread-like substances, assuming this is the nucleus, all these thread-like substances are chromatin. Now this chromatin, they consist of DNA also. So they include DNA that have already replicated. So this DNA in most cases must have sent information through these processes. So they have already replicated themselves, replicated. So when they become, they start to gather up, they start to condense, they start to gather up proteins in the nucleosome. So they start to gather up proteins. When they gather up proteins, they become visible. They start to gather up proteins. They become visible. Because they've replicated themselves. So one can replicate another. So when this one replicates itself, so those 2 that have replicated themselves can cross like this and meet at a center or a centric center called a centromere. So I just said there. So they can meet they meet at a center, at the center called a centromere. So they meet at the center. So in most cases, they are not equal. Like this is like equal equal halves. So the centromeres are the equal half. In some cases, the chromosome can look like this. The chromosome can look like this, where one of the chromosomes is actually, the centromere is not actually at the center. Okay, so when the centromere is at the center, it's metacentric chromosome. So they are very chromosome, telocentric, metacentric, and so on. So subsequent videos, not all information I can say here, but just note that this chromatin thread, they gather or they condense and form chromosome during cell division, in the prophase of cell division. So in the prophase, so for meiosis, prophase 1. So they gather up and form, they gather up to form, what's it called? They gather up to form the chromosome. When they gather up, during this gathering, they must have replicated in their interface. They must have so when they replicated, so two pair that have replicated each other, they cannot cross. Two identical chromosomes, two homologous chromosomes that have replicated each other cannot cross and meet at the centromere. Each of the so like this, they are called homologous chromosomes. So they are homologous. So the information here is information here. So the information here is information here. So now quickly, each of the chromosome or each of the homologous chromosome on the chromosome, each of two homologous chromosome. So how will I put it down? So this is an homologous chromosome. It's an homologous chromosome. Each of the DNA strand like this, each of the DNA strand of protein is called a sister chromatid. So this is called a sister chromatid. So each of the homologous strands is called a sister chromatid. A sister chromatid. So this is a sister. So two sister chromatid makes up an homologous chromosome. Two sister chromatid makes up an homologous chromosome, and the two sister chromatid are joined at a center called the centromere. They are identical. These chromatid are identical to each other. So these chromatid, they cross over during cell division. So chromatid are formed in prophase. Chromatid are formed in prophase. So they are identical chromosome molecule that cross to form an homologous chromosome. Okay, so each of the homologous chromosomes carry the same gene, same material, the same RNA sequence, same DNA sequence. Okay, for in prophase, you can have further having crossing over, where to have two homologous chromosomes cross over to enhance genetic variation. So genetic variation. So that's how it is for that. That's when we take chromatin chromosome and DNA. Okay, when we take chromatin chromosomes and DNA, and this segment can be called. This segment can be called a gene T. This segment can be called a gene for albinism A. Yeah, we will now say two genes can be formed on the same chromosome or the same homologous chromosome. Two genes can be formed on the same chromosome. So such genes are said to be linked. Yes. So those are non-Mendelian characters. We'll talk about them in subsequent videos. Okay. There are some terms in genetics that will be needed again. Before I quickly explain the RNAs and their function, there are some terms in genetics that we need. There are some terms in genetics that we need. There are some terms in genetics that I will need. Some basic terms in genetics that are needed. They include alleles. Yes, alleles. What are alleles? Okay, what are alleles? Alleles are they are what will I put it? They are traits with constructing characters. Okay, they are traits with constructing characters. So they are found on a chromosome. These alleles are found on chromosomes. They are the segment of chromosomes. Okay, they are the segment of chromosome. When they occur in pair, we call them genes. So chromosomes normally so these two. So on this two, so this one T can be found here, small t can be found here. So these two, so these two will have complementary genes. So each of the trait found on this chromosome is called an allele. It's called an allele. So alleles are traits with constructing characters. Alleles in pair make up what we call genes. Mendel already said that they are in pair and they can segregate. Genes are in pair and they can segregate. Okay, I'll talk about Mendelian characters in this video. So alternate form of a gene, it can be called alleles, or traits with constructing characters are called alleles. Then these alleles can be homozygous, or these genes can be homozygous or heterozygous. What does it mean to be homozygous or heterozygous? Homozygous simply means that the pair are usually the same. They're usually the same, and heterozygous means the pairs are usually different. So for heterozygous and homozygous traits. Simple. Now, what about there are two types of alleles? There are two types of alleles. An allele can be dominant, and an allele can be recessive. So what's a recessive allele? What's a dominant allele? Dominant allele is one that can be expressed in the presence of another. So in the presence of another trait, if you are expressed, you are dominant. While if you are not expressed in the presence of an other, you are recessive. So in theoretical genetics, we express dominant traits as big cap letters and recessive traits as small letters. Okay, which is not always so. So we have this mindset that tall traits will, for example, this big T is tall, this small T is short. That tall traits will always be dominant over short traits. In genetics, it's not like that. In genetics, a short trait can dominate a tall trait. But if you were expressed in the presence of another trait, you are dominant. But if you are not expressed in another trait, you are said to be what? Recessive. Okay? So you are said to be recessive. And recessive characters can only be expressed in homozygous condition. So once the only time it can be expressed is homozygous condition as a trait found on a DNA or as a segment of a DNA. The only time it can only be expressed is only where there's two of you, or it's only when you are complete in two. It means that you are said to be recessive. They are said to be recessive. So that's for allele. Now for RNA, I said I was going to talk about the RNA very, 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 very fast. Now I said the difference between the RNA and DNA is the nucleotide. Okay, DNAs are the ones that form RNA in the cell. Okay, DNAs they can produce RNA through a process called transcription. These are enzymes called RNA polymerase. Now RNAs can be three: tRNA, mRNA, and rRNA. rRNA are found in the ribosome. Okay, they're found in the ribosome. They help in protein formation in the ribosome. Now, what's the function of a tRNA? A tRNA, T means transfer RNA. tRNA help to transfer, what's it called? Materials from the nucleus. They have to transfer information from the nucleus, from the DNA. They are the ones that are produced, and the DNA produced. They have to transfer information from the nucleus to the mRNA outside the nucleus. They transfer information from the nucleus to the mRNA outside the nucleus. So the mRNA translocate this information to the rRNA in the ribosome, which help to further express this ribosome by itself in the ribosome by telling the type of proteins we produce. So these are the three types of RNA. mRNA stands for messenger RNA. rRNA stands for ribosomal RNA. Remember, we are still on the basis of this chapter. I already said the difference between RNA and DNA is their nucleotide. RNA and DNA is their nucleotide. Okay, so if genetics is a very broad aspect of science. So if you feel there's any aspect you have a controversial thought on, okay, this video is coming to a lot of extensive research. Now, if you think you have any aspect you have a controversial thought on, which I know there will not be if you really read, not thinking, read your books. If you really read, I don't think you have an issue. So just say it in the comment section. Any aspect. So that's any aspect. So the other terms in genetics, that we can talk about. They include things like talked about gene chromosomes. Talked about gene chromosomes. Yeah, these last two things I'll talk about is the phenotype and genotype. Almost forget about it. genotype and phenotype. Now, what are genotypes? What's a genotype? What's a phenotype? The phenotype is the physical appearance of an individual. So the sum physical appearance of every living organism is known as the phenotype. Yes, the sum physical appearance of every organism is called the phenotype. Okay, the phenotype is influenced by two things: the sum genetic material of the organism, which is called the genotype, and the environment. Okay, and the environment. Some texts normally say that the phenotype consists of the inherited character and the acquired character. So acquired character is environment, inherited character is genotype. So what's genotype? Genotype are all the genes inherited from the parent. All the genes, total genetic makeup of an organism inherited from the parent is the total 46 chromosomes. All the genes on the 46 chromosomes are known as the genotype of an organism. So with this, I'll be able to place an introduction on genetics. This series of video will continue for the next 2 or 3 videos. So I'm going to talk about genetics extensively. So we're going to be looking at genetics. I'm talking about genetics extensively. I'll talk about variation. So next video will be on Mendelian and non-Mendelian inheritance. Mendelian inheritance and non-Mendelian inheritance. So Mendelian inheritance has to do with Gregor Mendel and his theories. I'm going to explain. I'll talk about non-Mendelian inheritance, variation, crossing over. Then after this series, I'm going to do a series on cell division because we really need cell division. We really need to understand cell division, especially when it comes to this transcription, translocation, and replication. I will show you in interface. In interface stage, I will show you a video on mitosis. I will show you what this when these things occur in interface. So, but that's how it happens. Especially these two processes, these two. So it happens in interface. So that's how it was. So I remain the analogous until the next video. Bye. So God bless you. Thank you for watching. Make sure you hit the subscribe button. If you are watching, like this video. Like this video. See, if you don't like this video, watch your back. Watch your back. So see you guys. Have a nice day.